Here we can see the standard curves obtained with a variety of proteins and glycans. 2 biopharmaceutical samples were analysed using the OCTET platform: follicle-stimulating hormone FSH and an Fc fusion protein. The method used GAL1 and CA1 RPL-SAX sensors to detect terminal beta-1,4 galactose and sialic acid structures. The linearity of FSH and Fc fusion binding to either beta-1,4 galactose or sialic acid structures was determined by the binding rate of 2 replicates at 8 concentration levels, ranging from 500 micrograms per ml down to 7.8 micrograms per ml. The acceptable assay range was determined to be between 7.8 to 500 microgram per ml with less than 20% CV values. The blue dots indicate the standard curve points. We also ran 6 replicates of 200 microgram per ml to determine the repeatability and the precision of the OCTET method, and these can be seen as red dots on the standard curve plots. Overall, we saw high R-squared values and good percentage CVs at less than 20%.